good morning. Welcome to my AB Biology channel. Today our topic is Krebs cycle. You know very well, in aerobic respiration, Krebs cycle reactions take place. Krebs cycle reactions are related to aerobic respiration only. In aerobic respiration, there are two stages as earlier studied. The two stages are glycolysis and Krebs cycle. The Krebs cycle will not be in anaerobic respiration. In aerobic respiration only Krebs cycle reactions happen. In mitochondria, in the matrix of the mitochondria, Krebs cycle reactions take place. But for better understanding these Krebs cycle reactions, we have to go to the previous video where I explained the glycolytic process and also the link reactions. When you have that idea, these Krebs cycle reactions will be understood very clearly. So please watch my previous videos, link reactions and glycolytic reactions. How? Briefly, I am saying here, in glycolytic reactions, which is the first oxidation stage, the end product of the glycolysis is pyruvic acid. Two molecules of pyruvic acid are formed here. And those two pyruvic acid molecules get further oxidized. But as I told already, the pyruvic acid will not enter or participate in the Krebs cycle reactions directly. So, the pyruvic acid which is formed by the end of the glycolysis should be converted into acetyl coenzyme A. So, acetyl coenzyme A will participate in the Krebs cycle reactions. So, here I mentioned here this is glycolysis, and in glycolysis the glucose here, which is a substrate 6-carbon compound, is oxidized to two molecules of pyruvic acid. Pyruvic acid is a 3-carbon compound here. So, it is a enzymatic process, already you learned that. So, here the end product is pyruvic acid. In link reaction, the pyruvic acid molecules get oxidized. Before the entry into the matrix of the mitochondria, the pyruvic acid is oxidized to acetyl coenzyme A. Already here. So, this is a link reaction. Already I explained. Watch my previous video where you get the idea about link reactions. Pyruvic acid is converted into acetyl coenzyme A. Two molecules of acetyl coenzyme A here as two molecules of pyruvic acid or the end products of the glycolysis. So, this acetyl coenzyme A is a connective link between the glycolysis and Krebs cycle. And this acetyl coenzyme A going to be involved in Krebs cycle reactions. Here you see this is mitochondria. Okay, this is matrix of mitochondria where you will find number of enzymes which are responsible for the Krebs cycle reactions. The acetyl coenzyme here enters. This in the form of this acetyl coenzyme, this will enter. That point you have to understand. This will participate in the Krebs cycle reactions. That point you have to understand. Now, let us observe the Krebs cycle reactions. Krebs cycle reactions also enzymatic. What are those enzymatic Krebs cycle reactions? What is the yielding of the energy? How much quantity of energy is released in Krebs cycle? And how many molecules of NADH and FADH2 are formed? Okay. So, let us study about the Krebs cycle. This is the overall reactions of Krebs cycle. This is a flow chart representing clearly where a particular kind of enzyme is involved and where oxidation process happen and where do you find the release of energy. like this. And also decarboxylation process, where exactly will happen, and oxidative decarboxylation, in which step of Krebs cycle happens. So all these points you are going to learn. But starting with the Krebs cycle here, where do you find the Krebs cycle reactions? Those reactions occur, as I told already, matrix of mitochondria. So all these reactions were clearly observed by Sir Hans Krebs. Sir Hans. So these reactions also known as Krebs cycle reactions. Let us study about the Krebs cycle reactions. What type of reactions are there? How these reactions will take place in matrix of mitochondria. You know very well, acetyl-CoA is the connective link between glycolysis and the Krebs cycle. Here acetyl-CoA is a 2-carbon compound. We start Krebs cycle with acetyl-CoA. It is a 2-carbon compound. Okay. And this acetyl-CoA here, this combines with oxaloacetic acid, which is available in matrix of mitochondria. But as oxaloacetic acid concerned, it is the final product that is produced so that continuation of these Krebs cycle reactions will happen. Krebs cycle reactions will be continued with the formation of this oxaloacetic acid only. Shortly we can say that OAA. OAA. This is a 4-carbon compound here. Acetyl-CoA is a 2-carbon compound. This is the 4-carbon compound here. This is. And with the combination of these, see here, and citric acid is formed here. Citric acid is the first product, stable product of the Krebs cycle reactions. Stable product of the Krebs cycle reaction. This is the first and stable product. This is also known as tricarboxylic acid, as it is containing 3 carboxylic groups. Citric acid cycle or tricarboxylic acid cycle, TCA cycle shortly. But here, what is happening in this particular reaction? In this particular reaction, acetyl-CoA going to be combined with oxaloacetic acid. But actually here acetyl group is combined. Here CoA is released. CoA is released. So in the step of this reaction here, water molecule is added. You know very well, addition of this water molecule is nothing but a hydration process. Hydration. So this is nothing but hydration here. But here acetyl-CoA is going to be condensed with oxaloacetic acid. That point you have to understand. And this step is influenced by enzyme. The name of the enzyme here is citrate synthase enzyme. Citrate synthase enzyme. Okay. This is the first step of reaction in the Krebs cycle. And in the next step, here citric acid is a 6-carbon compound. That point you have to understand. You have to understand the number of carbons present in intermediate substances. But here 6-carbon compound. Again, this citric acid undergoes a particular kind of reaction catalyzed by this achotinase enzyme. Achotinase enzyme here is catalyzing this reaction. Here water molecule is released. Water molecule is released. So this is dehydration process. Dehydration happened here. So the resultant intermediate substance here is cis-acotinic acid, which is a 6-carbon compound. This cis-acotinic acid in the next step changed to or converted to isocitric acid. So here there is no change in the number of the carbons here. So it's cis-acotinic acid, isocitric acid containing the same number of carbons. But here one water molecule is added here. In the previous step, water molecule is released, that is nothing but a dehydration process. And here water molecule is added, this is hydration process. And then isocitric acid, which is a 6-carbon compound, undergoes oxidation here. Here you have to remember, very important step. conversion of this isocitric acid to oxalosuccinic acid. Why you have to consider this important step means here oxidation process takes place here. You know very well oxidation, release of these NADH molecules, NADH plus H plus molecules. How this will happen? Earlier I told you this NAD is a coenzyme. This coenzyme on receiving electrons. Here electrons are formed from the substance here. Okay, during this process of reaction, those electrons are accepted by NAD. Hydrogen atoms are removed. Hydrogen atoms are removed. This is dehydrogenation process. You learned that also. And as a result, NADH is formed. Here NADH is formed. NADH. This is oxidation process here. So this step is influenced by enzyme, that is isocitric dehydrogenase enzyme. Isocitric dehydrogenase enzyme. Then after oxidation, here what is the fate of the oxalosuccinic acid here? Now oxalosuccinic acid, here you see, this is also 6 carbon compound. It undergoes carboxylation reaction. It undergoes decarboxylation. Undergoes decarboxylation here. This is decarboxylation. Here this is oxidation. Here this is oxidation. This is decarboxylation. One carbon in the form of carbon dioxide is released here in this step, while oxalosuccinic acid is converting into alpha ketoglutaric acid. One carbon in the form of carbon dioxide is released, so it is a decarboxylation process. That decarboxylation process is catalyzed by enzyme decarboxylase. One of the enzyme here is involved which catalyzes the reaction, that is oxalosuccinic acid to glutaric acid, alpha ketoglutaric acid. But here one change will observe here. Oxalosuccinic acid is a 6 carbon here. One carbon is removed in the form of carbon dioxide here. Oxalosuccinic acid undergone decarboxylation here. And then here glutaric acid is formed. But glutaric acid containing only 5 carbons. Already one carbon is removed here. So alpha ketoglutaric acid is a 5 carbon compound here. And then what happens? So this is also catalyzed by one enzyme, important enzyme, that is what is called decarboxylase enzyme. This is decarboxylase enzyme here. And here in this process, what is happening? Here an oxidation process happens here in the conversion of this isocitric acid to oxalosuccinic acid. And also here the alpha ketoglutaric acid is converted to succinyl coenzyme A. Here also NADH oxidation process happens. So here oxidation process, here also oxidation process here also. Okay, but one point you have to remember here. Here along with the oxidation here, carbon dioxide is also formed, release of carbon dioxide here, that indicates what? Decarboxylation. Here decarboxylation only happened here in the conversion of oxalosuccinic acid to alpha ketoglutaric acid. But alpha ketoglutaric acid undergoes oxidative decarboxylation. Here both happen here. This is oxidation process, this is decarboxylation process. So together it is a oxidative decarboxylation. So that point you have to understand. So you will be asked one important question in the NEET. Where do you find oxidative decarboxylation in Krebs cycle? So you have to remember this step. In the conversion of alpha ketoglutaric acid to the succinyl coenzyme A. But here, very important point you have to remember. Here, alpha ketoglutaric dehydrogenase enzyme is involved here. This is one of the complex. Earlier I told you in link reactions. Please observe my previous video, that is a link reaction, where you will find what is about the dehydrogenase enzyme complex. The complex consisting many combination of many enzymes. Am I correct or not? Already I told you coenzymes are NAD here, NAD coenzymes, FAD like this, and also lipoic acid like this, lipoic acid, and also magnesium ions, cofactors. All these cofactors helping here. Dehydrogenase complex is a combination of enzymes, coenzymes present in these particular enzymes, which increase the efficiency of enzymes or activity of the enzymes. Those are NAD, FAD, lipoic acid. Already I told in link reactions. Please go to link reactions and observe what is happening in link reactions. Okay. And here the alpha ketoglutarate acid, after undergoing oxidative decarboxylation, forms succinyl coenzyme A here. But one point you have to remember, coenzyme A here is added. Coenzyme A added. One coenzyme A here is added, added here. So glutaric acid, alpha ketoglutarate acid converted into succinyl coenzyme A. Here one coenzyme is added. Okay. On receiving coenzyme A, succinyl coenzyme A is formed. But here succinyl coenzyme is a 4-carbon compound. Already I told you glutaric acid, alpha ketoglutarate acid is a 5-carbon compound. Here one decarboxylation happened here. Here also oxidative decarboxylation. Decarboxylation followed by oxidation process here. So one carbon is lost here in the form of carbon dioxide, and here succinyl coenzyme, that is 4-carbon only formed. But coenzyme added here. Coenzyme added here. And then in the next step you see here, these succinyl coenzyme A is here, which is a 4-carbon compound, converted into succinic acid. Succinic acid also 4-carbon compound here. In this particular step here, one of the enzyme involved, okay, that enzyme is known as succinyl thiokinase. What do you call it? Succinyl thiokinase. Okay. So just like how other reactions happen in presence of enzymes, the same reaction happens here with the help of the enzyme, that is a succinyl thiokinase. But here one water molecule is added. But you have to remember, very important, this is the step which is very important, you have to observe. Why? Because here energy release takes place. Energy always releases in the form of ATP, adenosine triphosphate molecules. In case of plants here, directly ATP is produced. Whereas in animals, form of energy is GTP. GTP. GTP stands for what is this? Guanosine triphosphate. Outward GTP is converted into ATP and it is made available. It is made available. But here, one important point you have to understand, directly energy is getting released from metabolites. From metabolites, directly energy is released. So, it is a substrate level of phosphorylation. Already, I told you, what is substrate level of phosphorylation? If ATP energy is generated directly from metabolites, then it is called substrate level of phosphorylation. So, you will be asked the question, where exactly the substrate level of phosphorylation? So, you have to remember this step. I mean in the conversion of what succinyl coenzyme to succinic acid. Here generation of energy directly takes place here. Actually, energy is released in the form of NADH and FADH2. NADH plus H plus and FADH2. Indirectly, energy is stored here. But here directly energy is released. So, it is a substrate level of phosphorylation. Then, the succinic acid which is formed here, that is converted into fumaric acid here. Fumaric acid is also 4-carbon compound. There is no change in the number of carbons. Okay, no carbon dioxide is released in this process also. Here also, this is also one of the important step here. Okay, here oxidation happens. But here not NADH molecules produced. Here, FADH2 molecules are produced here. FADH2 molecules are produced. FAD stands for what? Flavin adenine dinucleotide. This stands for flavin adenine dinucleotide. These flavin adenine dinucleotide, just like NAD, receives electrons which are formed during the reaction in this step. Those electrons are received by FAD and FAD gets reduced here in the form of FADH2. I mean here also one oxidation process takes place. This is also one oxidation. Oxidation. Here oxidation, here also oxidation process, here oxidation process, one more oxidation process here. Okay. So FADH2 are formed. Then under the influence of enzyme that is succinic dehydrogenase, here also enzyme that is called succinic dehydrogenase. Then fumaric acid, which is a 4-carbon compound, and is converted into malic acid. The malic acid or malate, it is also 4-carbon compound. Here water molecule is added. So it is a hydration process. Hydration process here. Okay. And malic acid is 4-carbon compound is formed. Then malic acid is finally converted into oxaloacetic acid, which is a 4-carbon compound. But oxaloacetic acid is repeatedly involved in Krebs cycle reaction. Continuously it will be formed. So here also the step is catalyzed by enzyme that is malate dehydrogenase enzyme. Enzyme is malate dehydrogenase. Here also an oxidation process happens. Oxidation. See here oxidation. Here also oxidation. Here oxidation in this step. Here also oxidation. There you see, in the conversion of isocitric acid to oxalosuccinic acid, oxidation process happens. But okay. So again these oxaloacetic acid will participate in the Krebs cycle. Okay. Important is the formation of citric acid, which is stable product here, which is the first product of the citric acid cycle. The cycle will be continued like this. Okay. So this is the overall Krebs cycle reaction indicating presence of enzymes and release of energy here in the form of ATP or GTP, and where exactly the oxidation process is taking place, and where do you find decarboxylation process. Okay, where oxidative decarboxylation happens, like this we have to remember. So this is very important. Once you have to understand once you have to go through this thoroughly. Okay, so what are going to be learned here? I mean, what you have learned from these Krebs cycle reactions here? How many but one point you have to remember here, acetyl coenzyme molecules is 1. Actually, by the end of this, I have taken only acetyl coenzyme A molecules only 1. Oxidation of 1 molecule of acetyl coenzyme A resulting in the formation of like this. But actually, by the end of glycolysis, how many number of acetyl coenzymes are formed? 2 molecules of acetyl coenzyme, not 1. 2 molecules of acetyl coenzyme. Am I correct or not? So you have to double the number here. I have taken only 1 molecule. This is account for 1 molecule. By the oxidation of 2 molecules of acetyl coenzyme A, here I am doubling citric acid 2 molecules. Here 2 molecules. And isocitric acid also 2 molecules. This is also 2 molecules. Okay, and 2 molecules of alpha glutaric acid. Here 2 molecules. Here also fumaric acid 2 molecules. Malic acid 2 molecules. Oxaloacetic acid 2 molecules. Okay, so we have to double the number. It's very simple. For the oxidation of 2 molecules of acetyl coenzyme A is a connective link which is formed by the end of these glycolysis here. Okay, 2 molecules of acetyl coenzyme A form. So after undergoing oxidation, 2 acetyl coenzyme A yielding this energy and also NADH at particular step here. Okay, that point you have to remember. So how many NADH molecules, where NADH molecules are formed, and how many number of NADH molecules are produced in Krebs cycle reactions. Krebs cycle reaction. So here I will write here number of NADH molecules. How many is here? This is you have to double it. 2 NADH here. Here also 2 molecules of NADH 2, and here also 2 molecules of NADH. Okay, how many molecules of NADH are formed? 6 molecules of NADH for the oxidation of 2 molecules of acetyl coenzyme. So NADH number is plus H plus NADH2R. This is 6. That point you have to know. How this is 6. Okay, for the, and how many molecules of FADH2 are formed? Where do you find the formation of FADH2? Here you see in this step. In the conversion of succinic acid to fumaric acid here. So FADH2 here is formed. But how many here? 2. We should double, no? Okay. So 2 molecules of FADH2. FADH2 molecules are 2 here. 6 NADH here. How many molecules are carbon dioxide here? Here also you see here. This is 2 carbon dioxide molecules here. And this is also, I will write another ink. here 2 molecules of carbon dioxide here, carbon dioxide here also 2 molecules of carbon dioxide. So how many molecules of carbon dioxide were formed? Carbon dioxide which are formed here, carbon dioxide number 4. Number of carbon dioxide molecules which are produced 4 here. Okay, 2 carbon dioxide molecules here, here also 2 carbon dioxide molecules here. Where do you find the decarboxylation here? Decarboxylation 1 here. I mean 2 decarboxylation here happened. Okay, 1 decarboxylation here happens. Okay, that point you have decarboxylation 1. How many oxidative decarboxylations here? This is 1 oxidative decarboxylation. At one place only oxidative decarboxylation happens. Oxidative decarboxylation here, the one here. So decarboxylation 1 here, deoxidative carboxylations 1, number of carbon dioxide 4, and number of NADH2 molecules are 6, and number of ADH2 molecules are 2 here. This is a very important discussion, especially this is useful for NEET students. So you have to go thoroughly through all the reactions where exactly a particular kind of reaction happens, yielding NADH, FADH2 molecules and generation of energy. Okay, all this information should be studied. Okay, thank you very much for watching my video. Okay, please like, share and subscribe.